In interphase, the cell spends most of its time replicating DNA and performing normal cell functions. Once the DNA in the cell has replicated, the cell is ready to divide. There are now two copies of all the genetic information in the nucleus of the cell, and the red and blue yarn represents the sections of DNA, and that messy part that you see is the chromatid.
In prophase, the DNA winds up into organized structures called chromosomes and become visible. Each half of a chromosome is called a chromatid and contains the same genetic information. The spindle apparatus begins to form and move towards the middle.
In prometaphase, the kinetochores are attached to the chromatids, which will eventually move those two chromatids towards the poles within the cell. And then that would lead into metaphase, which is where the membrane surrounding the cell, or I should say nucleus, breaks down, and the spindle apparatus extends fibers that attach to the center of the chromosomes. As the spindle fibers tighten, the chromosomes line up in the middle of the cell. And with metaphase, the spindle apparatus needs to extend the fibers to attach to the center of the chromosomes, which is why the nuclear envelope disappears before the cell divides.
In anaphase, the spindle apparatus pulls the spindle fibers towards themselves, causing each chromatid to pull away and separate to opposite sides of the cell.
In telophase, now that the chromatids have separated, the cell will start to form a nucleus to protect the DNA. The spindle and spindle fibers release the chromatids and begin to be broken down by the cell. The cell begins to split its cell membrane and cytoplasm in half to begin the formation of two cells. And does each side of the cell have the same DNA? Yes, because the two cells that would be formed later on would be identical, and the chromosomes are daughter chromosomes.
In cytokinesis, the cell has separated and formed two completely new cells. The chromatids unwind back into chromatid, so the DNA can be used to create proteins. Each cell goes back into interphase and starts replicating the DNA to prepare for another round of mitosis.